So those were some comments on the acute leukemias in the lymphoid lineage. That's acute. That's within the primary lymphoid organs. Okay. But today is about secondary lymphoid organs. Okay. And it's about nodes. So today it's about lymphoma, and you should imagine lymph nodes mainly, or extranodal lymphoid tissues or organs. Okay. Till now we talked about the lymphoid lineage, the acute leukemias. Those were lymphoproliferations of precursor cells, okay? So it's the lymphoproliferation of immature cells. And as we talked about, we got only one entity, and it's the ALL/LBL. But then we have versus lymphoproliferation of mature lymphocytes. Okay, so those could be T, B, or NK. But we won't talk about NK, that's much rarer today. And these are, of course, lymphomas. And of course, you can call it also leukemia over here, but we don't use the term acute, that's important. Okay, so it's rather chronic, of course, chronic leukemia. And then the boundary in between these two, it's blurred. If it's more like solid, you call it lymphoma. If it's rather liquid in the blood, it's leukemia. But typically you put it together, although there are some special types we're going to talk about it, which are rather only liquid, and the other ones rather solid. But anyways, so today it's different. It's not about bone marrow, it's about peripheral lymphoid tissue, about the secondary lymphoid organs or tissues. And why it is so common? Because this is the most common one. The lymphomas are much more common than leukemias. And out of the lymphomas, we have the two types, the Hodgkin and non-Hodgkin type. And the non-Hodgkin type, of course, is more common. Okay. Well, first of all, we're talking on lymphomas. So what you should imagine is a lymph node. It's a lymph node. And why it is so common? Well, typically, you should always imagine some kind of infection, some kind of irritation, some kind of chronic inflammation. You should imagine viruses. And why? Well, the reason why is because over here in the lymph node, or follicles, which could be located in extranodal lymphoid tissues as well, that's the place where the somatic hypermutations happen and class switch recombinations and clonal expansion. So over here you should imagine a germinal center, the germinal center of a follicle, and that's where everything happens. Because like physiologically, we want our naive lymphocytes, and we're talking about the B line over here, to develop some affinity to antigens. And as you know, these processes are done by changes of DNA. So that's why the longer you have inflammation, the longer you have some kind of irritations, and the more you have antigen presentations, well, there's a higher likelihood that you're going to develop something which is uncontrolled, so lymphoma. So what happens over here in the follicles? Well, of course, so somatic hypermutations. Okay. Class switch recombination, which is actually what happens in the class switch. It's a massive change of a chromosome. Although it's not translocation, that's important, it's intrachromosomal recombination. So intrachromosomal recombination, okay? But it's a manipulation with your chromosome, so it's massive. And of course, there's a clonal expansion. All of this is physiological, but unfortunately, the more common this happens, the higher the probability that this will get dysregulated and some kind of translocation, which is pathological, happens, for example, okay? So this is the reason why the lymph nodes, and of course, as you get older, you have higher probability to develop a lymphoma. So over here, we can see some mechanisms. So let's sum up the main mechanism. Let's go through them, okay? So I'm gonna clear it out. Wait a sec. Okay, now, and what are the mechanisms? Mechanisms that increase the risk of lymphoma development, okay? So first of all, this one is connected with the interaction of viruses and a lymphocyte. So there could be a direct oncogenic mechanism. There could be a direct, and I'm going to put it in red. So there could be a direct oncogenic mechanism. Well, this means that the virus, and there are many viruses I'm going to mention, and they don't have to be integrated in the DNA of a lymphocyte. They just can be present there as a circle of DNA, whatever. And this presence, of course, somehow interacts with the lymphocyte DNA. Okay, so it can like change gene expression, it can modulate host chromatin, transcription, whatever. So only the presence of the virus DNA that doesn't have to be integrated in the DNA makes mess over there. This drives the lymphocytes towards developing a lymphoma, which is crucial. Of course, then we have retroviruses, and this is pretty obvious because they are typically incorporated into the lymphocyte DNA. Then the another mechanism, which is really important, and the main one, like in general, it's a chronic immune activation. Okay, so chronic immune activation. And this is a very general process, but basically what it means that there is some chronic process, and it could be inflammation because of again, virus, there's a chronic infection, viral infection, or bacteria, doesn't matter. So any kind of irritation, if there is a local immune reaction that is drained by a lymph node, and this lymph node is stimulated, it's activated, and if the process is chronic, this can lead to, again, lymphoma induction. Well, and the third mechanism, the major ones, is immunosuppression. Immunosuppression. And this is, again, this happens in the follicle where you have, for example, the T helper follicular cells, the CD4s, which are supposed to maintain, and they're supposed to help with the maturation affinity. And if there's a decrease of them, yeah, of course, this control fails, and then you have, again, increased risk of let's say, malignant clone developing over there. So what are the viruses? I'm going to mention a few, but of course, the most important and very common and associated not only with lymphomas, but also, for example, with multiple sclerosis. Now we know it's EBV virus, okay? So it's a herpes virus. We're going to talk more about it a bit. Herpes virus. But this one stays in the cells of the lymphocyte and interacts with the transcription machinery and everything. And if it's chronic over there, it leads to very commonly lymphoma development. So there's a major connection. And if it combines with other viruses like HIV, this is even more increased. EBV, it's not integrated in the DNA, but as I said, it interacts with the DNA machinery, but it also, of course, causes a chronic inflammatory stimulation. So let's say antigen stimulation, etc. Okay, so it's very, very important. And we're talking over here about B lymphocytes, and of course, this then has a important connection with Burkitt lymphoma. This is classic. But then also diffuse large B cell lymphoma or Hodgkin lymphoma. Okay. Another important virus that is connected with lymphoma is hepatitis B virus. This virus integrates into the DNA, but of hepatocytes. It's very rare that it would be integrated into lymphocytes, but over here the main mechanism is, again, chronic immune activation. Okay, so chronic immune activation. So there's a chronic inflammation, and this can lead to lymphoma. Typically leads to diffuse large B cell lymphoma or marginal zone lymphoma. Okay, another example is also hepatitis C virus. This one, again, it's not integrated into the DNA of a lymphocyte, no. But again, the main thing is the chronicity and chronic antigenic stimulation. So again, there's a chronic inflammation. So chronic antigenic stimulation, this stimulates the follicles, they react to it, and this again increases the likelihood of lymphoma development, again in this case DLBCL or marginal zone, to make it simple for you, zone lymphoma. Okay, and another very important virus that is connected with lymphomas is HIV. So it's a retrovirus. Over here, it gets integrated, but actually not in the B lymphocytes, but T lymphocytes. And as we talked about it, this is integration in this case, but the lymphoma connection very much with the T follicular helper cells, so CD4 positive. It gets only to the CD4 positive cells. But what they cause, well, actually, it's again chronic stimulation, so chronic immune stimulation, but over here, plus immunosuppression, because the function of the T helper cells in the follicles, of course, is impaired. So there's a germinal center dysfunction, and this leads to lymphoma development, okay? Because the T lymphocytes, which are guards over there, and they're getting rid of the malfunctioning lymphocytes, B lymphocytes, that are not having optimal affinity, antigen affinity. So now they're lacking. This is a dysfunction, and this increases the risk of lymphoma development. So it causes, at the end, germinal center dysfunction. So this is one of the examples of immune activation and also germinal center dysfunction. Okay, so germinal center dysfunction, and HIV is again connected with many lymphomas. First of all, so diffuse large B cell lymphoma, but also, of course, Burkitt. We're going to talk about it again. And again, for example, Hodgkin lymphoma. Okay, and why am I talking about this? Why am I talking about viruses and germinal centers? Well, because first of all, the lymphomas, the B lymphomas, B cell lymphomas, they are associated with germinal centers. And that's why we can divide them into pre-germinal center, germinal center, and post-germinal center neoplasms. So that's one thing. Another thing is, why am I talking about somatic hypermutations? Well, there's a big difference between a B and a T line, because the T lymphocytes, they know nothing about somatic hypermutations. Once they leave the thymus, it's done. Their receptors, they undergo in the thymus the VDJ recombination, and then they are stable. They don't change in lifetime, in life of the lymphocyte, the T lymphocyte. Okay, that's in a big contrast to B lymphocytes. So that's why also the mechanisms through which the lymphoma can develop is different between B line and a T line. Okay, there are different mechanisms, different patterns. So that's what I want to stress next. I'm going to still repeat the acute leukemias, and I want to compare it with the B and T lymphomas, and I want to stress different mechanisms and also the, let's say, physiological processes that are the sites of impairment and that they at the end lead to lymphoma development. So let's break it down. So thanks for watching. And don't forget to subscribe and ring that bell. And as always, check the description below for supplementary questions and other materials.